Okay, so it's my pleasure to introduce our speaker for today, Dr. Vega, who is an assistant professor in the Department of Biology here at Emory. They got their MS in quantitative ecology and resource management from the University of Washington before getting their PhD in molecular and cellular biology and biochemistry from Boston University. They then went and did a postdoc at MIT and have been at Emory since 2018. Their lab focuses on the ecology of microbial communities using the model system C. elegans. So welcome, and the floor is yours.
Okay, thanks so much. All right, so before I get started, I just want to start by acknowledging people who do the work in the lab. So this is my group. I don't have a picture of Claire up here, but my graduate students, one of my undergraduates, C, and down here's Megan Taylor, who is now at the CDC as my research technician. She did some of the work that we're going to be talking about. Yes.
Share your screen.
Oh, hold on. All right, is that better?
Yes.
Stop. All right, there we go. Okay, so as you know, I work on microbiomes, usually host-associated microbial communities. And some of the questions that we're interested in, I like microbial communities because they're complicated. These are diverse communities with a lot of moving parts, with a lot of heterogeneity from instance to instance, and inherently they're a multi-scale system. We have interactions within kind of trophic level among microbes and migratory system. We're receiving migrants, we're losing composition over time to drift. And we have interactions with a host environment, which is moving at a different spatial scale and a different time scale. So these are problems that I really like. I like the composition of these problems. And the way that we get about these problems, because there are so many moving parts, it's very helpful to be able to take them apart. So we're an experimental and computational group, and we use a combination of bottom-up and top-down experiments and computational models, as well as database modeling to understand what's happening in the simplified systems at varying levels of simplified that we work with, so we can understand the rules that shape these bigger, more complicated, naturally occurring ecosystems that we don't have as much control over. We don't have as much access to what's happening in these cases. And the goal here is, because we are an experimental group, we're using tractable systems. What we want to do is create good data to inform the creation of good models. So the data that we're trying to create are really large multidimensional data across multiple conditions, across ranges of conditions that give us an idea of how patterns change across gradients. In this case, this is a piece of what one of my collaborators called the data cube. We have microbial community composition in C. elegans across. I think this is a subset of the many genotypes of hosts that we have in here. Here we're looking at what are the effects of host genotype across a range of things that we know are different across different kinds of hosts. What does that do to community composition? What does that do to community composition over time? And time is an important component here. So we have different types of hosts, we have space within hosts, we have the evolution of these communities, the dynamics of these communities over time, these time series type information. This is where we can actually get at the dynamics of these systems. The good models that we're trying to produce are models that describe and predict the patterns that we see in these systems. That can describe not just the trends, the central tendencies of the data, but the variation, the shape of the heterogeneity that we observe across instances of the same kind of community, the same kind of colonization. In this case, this is even just like singles views colonization. We see a lot of heterogeneity. Where does it come from? We look at multi-species communities that are colonizing the same host, different types of hosts, with a great deal of variation. Where does this come from? How can we generate models that can predict the patterns that we actually observe? Well, we're going to go back a few steps first. How did we get into this line of work? So the idea of processes across scales, of multi-scale processes, is something that has been fascinating to me for a really long time. This dates back to really when I was a kid and read Silent Spring. Has anyone here read Silent Spring? A decent number of nods. So this is really the first book that I read about ecology. And it's really fascinating for those who haven't read it. This book made a really big difference in how pesticides were regulated, were thought of in the US and globally. It's a book about about pesticides, and insecticides, these tiny clever chemicals that are used to control things that we don't want, and how they interact with the world around them. And so there's a lot of stories in here of accumulation. We spray these chemicals onto fields and they get into water and they get into algae and they get into fish, and those fish get into herons and get into eagles, and now the eggshells are losing their integrity. So there's a lot in here about the webs of life, the interconnectedness of things and the consequences of the interconnectedness of living things in the real world. That was just absolutely fascinating. And a few years later, now we can use math. Has anyone read Chaos? Okay, Lance, I'm not surprised Lance has read Chaos. So this
Yeah,
is,
sorry, I had to.
you did, it's required. It's actually required.
No, I recommend it.
I recommend it. Well, it's a really good book, and this is a popular science book. It's, you don't, like, I read this when I was 13. You don't need a lot of math to read this book about the emergent complexity of systems and how we can use math, how we can use how we can mathematically define rules or create models that reflect the rules that drive these big interconnected systems. We can use these models to understand what's going on, to describe the patterns that we observe. That's what I did. Went to University of Washington, Seattle, joined the core program, and got trained as a modeler. Now, I was working in field ecology at the time, studying dogfish sharks, and field ecology didn't work for me for a number of reasons. So I ended up as an experimentalist in Boston University in Jim Collins' lab. And Jim Collins, one of the things he's really interested in is heterogeneity in antibiotic response. And so I had a longstanding interest in heterogeneity, in the variation that we see in living communities and where it comes from. And so the project that I worked on there was on heterogeneity in antibiotic response, specifically in intolerance to antimicrobial treatment. So this is an experiment from my first paper. We've got a community here of E. coli, just E. coli in a microfluidic chamber. We've let them grow for a little while. And then for a few hours after they've grown up, we put in an inducer. So these are the cells, this is fluorescent channel, and we can see when we put in an inducer, there's a small molecule they produce as indole. It's a small molecule they can use as kind of a signal of impending stationary phase, or sort of can turn on stress responses. We can see there's a lot of variation in response to the signal. It's just flowing in liquid. We have some cells that respond a lot, some cells that respond a little, some cells that seem to respond not at all. Then once we give them a chance to respond, we hit them with an antibiotic. In this case, ampicillin, which is a lytic antibiotic, so they're going to explode if they're susceptible to responding to the drug. And we can see they're starting to bleb out and starting to explode, but the brightest cells are the ones that stay. And so we can see here the heterogeneity in response to a signal that has consequences for the survival of the population. But this is in my fluidic chamber. And real microbes tend to exist in not microfluidic chambers. So we want to put this into a host, enter the worm. So this is a point at which I started working with C. elegans as a model host system in which to look at the effects of signaling, effects of heterogeneity, specifically for antibiotic treatment at this point. And the reason we started working with worms is that at this point, basically, I needed a microfluidic chip with face. I needed something that was a responsive biologic environment where I could make a lot of them at once, that was repeatable, but have the biological complexity of a host. And so worms are great for that. You grow them on petri dishes, on lungs of E. coli. You can have thousands and thousands of them on an individual plate. They're self-fertilizing hermaphrodites. So you put down one worm and just get thousands and thousands of worms that are of the same genetic background. They're self-fertilizing, not selfing, not clonal. So there is some heterogeneity, but it's a very limited gene pool when you're only working with your own gametes. And because they're being raised on one plate, or placing made at the same time, the life history is very uniform. They're exposed in the same environment over time. We can synchronize them to get them all to be the same age, same stage, same size, all the way through our experiments. And as a result, they're transparent. So we can put bacteria in them that are fluorescent, or we can put reporters in them that are fluorescent, and we can see what's happening in the bodies of these worms. And importantly for us, because I'm not actually a geneticist, there's a bunch, you can call up the CGC and say, give me all of these mutants, and they will. And it's like $10 a strain. They're fantastic. They're also pretty easy to get a microbial community inside. So worms are bacteriivores. They're about 1 millimeter long, and they feed very efficiently on things that are roughly E. coli sized, give or take. And they'll just kind of move face first through their environment, eating anything that basically fits. As they do that, these bacteria get pushed through the pharynx, which is the mouth parts of the worm, into the intestine through this thing. This is a grinder. It's There's a cross section, so we're looking for cross way through. It's 3 chitinous wheels, like cog wheels. And when the worm swallows, they do this. Most of the bacteria are physically crushed by the mechanical action of the grinder, but a small fraction will survive, and those can then adhere to the intestinal epithelium, form biofilm, and now we have microbial. So we can take these things because they're small, they're tractable, and they don't mind what you do to them for the most part. We can take them and we can make the very well controlled experiments that we need to control what sources of variation are contributing. We can take worms from a single genetic background for any given genetic background, synchronize them so we're getting adults. We can clear out any bacteria that might have accumulated during growth, and then we can colonize them with whatever bacteria we want, with whatever mixtures we wanted to in well-mixed liquid culture, so they're encountering a uniform environment, which is a nice simplifying assumption, and then we can pull them out individually and measure what's happened in individual worms. And this gives us ways of snapshotting whole populations and what are the communities that we've gotten under whatever conditions it is we've imposed. And even under these very simple conditions, so it starts really early on, so still goes back to my PhD, we see a lot of variation in colonization across individuals. These, again, isogen-based individuals from the same genetic background in liquid culture, colonized at the same amount of time on a uniform mixture of bacteria. In this case, it's two bacteria, red bacteria, green bacteria. And we can see there's a lot of variation. So this is two color plus bright field image. We can see like this worm right here. It's yellow, it's basically yellow. It's got a lot of red, it's got a lot of green. Worm down here, very green, not a lot of red. Basically green worm. This worm up here, it's got a lot of red, basically no green. There's a whole lot of variation. Again, it's basically the same worm. So where's this variation coming from? So here's the basic problem that we're dealing with. We have a bunch of worms. Hosts are different from one another. Here we can control how different they are from one another to know they're the same genotype. We'll mix this up, but let's say they're the same genotype, but maybe the hosts are still different from one another somehow. And they're colonizing from a meta community of bacteria where we've got just green bacteria here, but we know that even within green bacteria, all bacterial cells are not the same. And we're usually encountering more than one kind of bacteria. So we have now different types of bacteria, each of which has heterogeneity within it. And these hosts, which are different in ways that we cannot at this point quantify, we know they're there, are going to acquire migrants stochastically from this metapopulation, from this pool of potential colonists. And within each of those hosts, now processes are going to start to happen. You're going to make more of yourself, you're going to die, and especially for early colonies, this is very stochastic. Birth and death are given stochastic processes. They're also deterministic processes where you have a growth rate and a death rate, which might be different for different bacteria, probably are different for different bacteria, might be different for different subtypes within a bacterial lineage, might be different across different individual worms. Now it's starting to get complicated. And then this is the data we actually get. We have the community composition in individuals. We have the difference in community size between individuals. And we need to try to dissect out what are the sources of variation that are contributing to the patterns that we observe. And this is where that tractability really comes in. We can start taking out sources of variation one by one, adding them in one by one to figure out what's contributing to the heterogeneity we see by changing the patterns of variation. And we need some expectations from what we see. One of the simple expectations that we like to use and refer to over and over here is a neutral model. It's just a neutral model of biogeography. And this is a stochastic model, assuming that you have a metapopulation, a pool in this case of microbes, that's well mixed, and some islands. Those islands are going to receive migrants stochastically from this metapopulation. All islands are the same island. They all have the same chances of receiving any given migrant, and all migrants are the same. They have the same chances of getting to any island, and when they're there, they take up one space. Each island has a number of spaces. Each migrant takes up one space. Once you're on the island, you will play by the same rules. All migrants are the same migrants. They have different labels on them, species label, whatever we're calling them, but they're the same migrants. They take up one space. They don't interact differently with one another. All the rules, once they get onto the islands, are the same. And what this leads to is an equilibrium that's defined by the combination of these stochastic processes, defined by the nature of the metapopulation and the relative rates of death and birth and immigration. This gives you the distribution of states that you expect over a given number of islands. So some of our previous work, my postdoc at Physical Existence at MIT was using the neutral theory of biogeography to see what happens in worms. So if we actually make neutral worms, can we get neutral biogeography back out? In this case, we're colonizing a bunch of worms with two species, two colors for simplicity. E. coli red, E. coli green, they're otherwise the same. And migration is one of the not, we can't really control birth and death too much. That's kind of property of the system. We control migration. We control migration by controlling how much bacteria we give the worms. So we give them a little bit of bacteria, we have a low migration rate, and this gives us this very kind of bimodal distribution. As we increase the amount of bacteria we're putting in, we start to get, we go from this uniform distribution to centrality, where worms start to look more and more like one another. We can see this in the images as well. So worms that are colonized at low densities have these very patchy colonizations. So say we have one migrant that enters and grows and eventually gets interrupted by something that doesn't have much space left. Some worms are basically all one color. A lot of worms are basically all one color. And then at high densities, because we're getting a lot of migrants, they're landing next to each other and we get basically yellow. We get these very well mixed communities with very short characteristic patch lengths. And if we compare to the expectations from theory, it works pretty well. As we change the migration rate, by changing CFUs, we transition from this bimodal regime down through kind of uniform into a unimodal regime. It works, it's biogeography. So a host can be an island. This host can be an island. But can it be a host for microbiome? This was E. coli red and blue. Very simple situation. What happens if we start making things more complex? We start adding differences in different kinds of bacteria. And it's not going to stay still. We know that things are going to change over time. Bacteria are great at changing. So how does the host shape, once we have community, how does the host shape evolution? How does adaptation happen? And what does it look like when we have adaptation of a community? So we start at the top. First, is this actually a microbiome? Does a worm form a microbiome? We have some working definition of microbiome that tells us some things that we need to look for to decide whether this is a microbiome or not. So we need a community of dynamically interacting microbes, a community more than one player dynamically interacting. There's the interactions. Those interactions need to give us patterns. And there needs to be characteristical ecological features. There are things that are true of microbiomes, like long-tailed species abundance distribution, there's relationship between mean and variance of abundance of species. And these are things that are true of communities generally and have a characteristic shape for microbiomes. We have some native bacteria. This was a few years ago. There were a couple of papers trying to investigate what is the native microbiome of C. elegans, because no one cared for like 50 years. But now we've got a couple of papers of when we actually find these things in the wild, what are they associated with? So the Sturzenstahl paper, Heinrich Schulenberg's lab, went out into a French-German fruit orchard, found some C. elegans, brought them back in and cultured some stuff. The original microbiome, native microbiome, covering most of the diversity they saw, that they could culture in the laboratory with 16 strains, which is what we have, and we picked 8 of those. This is the subset of 8 strains that we picked to basically cover the overall diversity that was found in this study, and to have colonies that we could distinguish from one another in agar plates, so that we could count data out of these, and that's going to give us some advantages. Now we have actual abundances. We're going to do this the same way we do our usual experiments. We're going to take synchronized adult worms, just wild type worms in this case, make them germ free, and we're going to colonize them on mixtures of all 8 of these species at 1:1 to 1:1 ratio. So uniform, well mixed meta community, nice simplifying assumption. For fixed periods of time, we'll refeed them periodically to make sure that we're forcing the meta community so it kind of stays still. And then at intervals, we're going to take out individual worms, bleach off the surface, grind them up, plate them out, want to see what's inside. Megan was a real trooper on this one. So this is just the N2. There's a whole bunch of these data. This is just N2. So N2 crystals are canonical wild, like laboratory wild type C. elegans. And here is a figure of 164 individual N2 wild type worms representing 10 experiments over about 18 months of time, where we have total abundance of each bacteria on the y-axis, and each of these is one of our species. Then each of these lines is the abundance of this bacteria in one worm, over 164 worms. It's a lot of data. We can see some trends. First off, there's some bacteria that are just super prevalent. So over here, like MIB71, we can see has very high peaks. We have some that are up at 10 to 5, 100,000 CFU per one worm. It's a lot of bacteria into a worm. There's a lot of variation. Some of them are down at 3 logs to 10 to 3,000 bacteria per worm, but it's present in 100% of worms, usually at a high fraction of the population. Then we have some bacteria, so MI27 is Arthropactrum, present at slightly lower maximum levels, still a lot of variation worm to worm. And then some stuff kind of down here off the end. So two species, Crosiobacterium and Acerotrophomonas, that just, they don't colonize very well. They're not there very often. When they are there, they're low relative abundance. So we have a shape of this. There's a lot, and there's a lot of variation between worms and across all taxa, across taxa considered as a whole. But there is a pattern, and we can take an average relative abundance across worms, and it shows you what you think it's going to show you. Where MYB71, which is an Ochrebacterium, worms love Ochrebacterium, is on average the majority of the population. Then there's some of these medium abundance, and these last two, Bacillus, Crosiobacterium, kind of falling off the tails. And we can see that it changes over time. So if we look, this is mostly after 4 days of colonization, refeeding every 2 days to kind of force the meta community. We can also take one after 2 days or 4 days or 6 days, and we can see there's movement in these communities. There's some sort of change that's happening, but the change doesn't have a uniform velocity. Day 2 and day 4 are almost offset, but day 4 and day 6 kind of overlap. So we started to slow down. We're starting to converge to a set of states. So this composition is what we can see. We start breaking this down. So does this actually look like a microbiome? So one of the things that we expect in a community, kind of broadly, is this Taylor's law process, where should be a power law relationship, log relationship between the mean variance species abundance. Michael Martini and Ilya's lab is doing some of this analysis, and we see something that's not too far off of a Taylor's law. With 8 species, it's pretty good. Further, we can look into what sort of interactions do we have? What sort of evidence do we have that there's interactions between players in the system? So, done this a couple of different ways, but one of the things that Michael did was to copula transform these data to try to extract the mutual dependencies. And in this model, a neutral model, the data should have zeros in all the off diagonals. We should have no dependencies among species because there's no actual interactions among species. Everything is 1 to 1 to 1. In this case, we have a decent number of terms that are significantly non-zero. Now, what this means is, of course, the question, because these are not interaction coefficients. These are informational dependencies. So this, and it turns out we try to actually fit a model to these, try to figure out, where could these coefficients come from? It's actually really hard to tell from snapshot data what these interaction coefficients mean. It could be that we actually have interactions among bacteria that are in some way generating these terms. We could have correlated parameters of bacterial host interaction. If different hosts are drawing parameters from sets, and these parameters are correlated across bacteria, you can also get these terms. Both could be true. We can't tell. We can't tell from these data. So we can take advantage of the tractability of the system and say, okay, let's try to independently estimate some things. So one thing, we were colonizing with 8 species. What if we colonize with each species individually? That way we can extract the interaction terms, birth, death, etc., from the single species trajectory, and what's left is the interactions. Okay, it's an idea. So it's work that Sachin's been doing in my group. So we take worms and we're exposing to 1 bacteria at a time. In this case, let's say it's Ochrebacterium, which is our most prevalent species. Now, we look at early times, we should have low total abundance. Maybe we've got some zeros here, it's a stochastic process of colonization. Over time, we should start to get growth, exponential swamping. We'll start to get everyone colonized. And then in sufficient time, we should start to see saturation. Everyone should be getting towards the capacity of the worm. When we do this, so this is MYB71, is what this looks like. We have a lot of variation, obviously, but this has the shape that we think it should. We have a lot of zeros and some low values at early time, about 3 hours of colonization. As we get to 1 or 2 days of colonization, most all worms are colonized. We're starting to get toward like 10 to the 5 maximum CFU, which is almost what we saw in the single species. So we can essentially worms group into one, or bacteria group into one of these types, where either we get this kind of Lotka Volterra like saturating response. 4 of our bacteria do this. We have growth, we have saturation at some level that's relatively high. Then we've got a couple of bacteria that don't. They're just kind of there. And so these are essentially our migration force bacteria. When they come in, there's a level that's basically maintained by migration for the most part, and doesn't look like birth and death are probably pretty close to one. So then what can we say about interactions? Now, this is a very simple model, and this is not correct, but just to give like a simple null that we can use for expectations. If, let's say, all worms have a given carrying capacity, if we're monocolonizing, each bacteria is going to take all the space available to it. If we are, say, colonizing with 8 species, on average, assuming all bacteria have access to all the spaces, each bacteria will then take 1/8 of the space that's available to it, and on average. And obviously, that's not exactly true, but the expectations get complicated. We can use this as a symbol though. So flat neutral model, we expect to get like a 0.9 log decrease, going from single species to multi-species. And sometimes that approximately happens. So B71 or good colonizer actually decreases by about 0.7 logs. 53, another pretty good colonizer, by about 0.9. Sometimes we get actually a lot more decrease. 2 logs, 1-2 logs. And sometimes we don't get a decrease at all. Some do better in multi-species. So it looks like there's probably interactions. Again, specifically what those interaction terms are, is still going to be difficult to infer as it turns out, but it looks like the evidence supports that there are interactions that are important for the structure of these communities. And further, mentioned briefly, there's heterogeneity across hosts. We can look at that. So for some kinds, so let's look at MIP27 or for background. After 48 hours of colonization, we see exactly about 2 logs of change, about 10 to the 5 to about 10 to the 3. 2 logs of variation total counts. And again, there's nothing else happening. There's just the same worms and the same bacteria in the same community, both at the same time. Sometimes we have like 4 logs. So not only do different individual hosts interact differently with the same bacteria, the pattern of heterogeneity changes across bacteria. All right, so we're in the process of working this into an actual model, but at this point we can say that this looks like all the terms for microbiome are being met. We have communities of dynamic interaction microbes that are interactions that are dynamic and stochastic, a lot of variation. We're working and figuring out the distributions. Turns out it's not trivial, that nevertheless have characteristic features. We can characterize central tendencies, we can characterize variations, and we have distributions of taxa that look like a microbiome. They're nautical from microbial community type ecology. As we said, these aren't standing still. This was a native worm microbiome, but to what extent does that mean that it's host adapted? What does host adapted mean? What does host adapted look like? So we started wondering, can we make a community host adapted? What does that do to the properties of the community? So starting with the set of bacteria, these have the same labels as our minimal community from the native worm microbiome, but these are from a soil collection. These are from the USDA NRL soil collection. They're from different places around the country, different soil samples, different agricultural sites. As far as we know, they've never seen a C. elegans. As far as we know, they've never seen each other. So they're really just around. And from these, we have 7 positions. We dropped one because at the time we didn't have a taxonomic ID for it, so couldn't include it. We've got 7 positions left. And at 3 of these positions, we have options. So we've got 1 strain representing Arthropacter, 1 strain representing Bacillus. We've got 2 strains representing Chrysobacteria. So this allows us to create 12 combinatorial communities with the same taxonomic positions, but slightly different strain membership, and all 11 single species isolates. And we're going to put worms on them, and we're going to passage them every 6-7 days for 9 to 10 passages and look at what happens to the communities that we're seeing inside these worms over time. What we see in terms of composition is what you tend to see in these types of experiments. We have this kind of fast, slow compositional change where anytime you take a community and put it into a new environment, there's going to be kind of a rapid kind of filtering change, and then there's going to be this slower change over time. And that's what we see over here. We have this kind of rapid initial change, and then by like the last couple 4 passages, they're going to converge. Specifically, we see that we tend to get to a state where Chrysobacterium, CX here, which is an allelopathic bacteria, it has it's bright orange, it secretes toxins, tends to kill other stuff, and it tends to kill the worms, tends to dominate these communities over time. And specifically, we have, so here's the whole dynamics, past 2 years, this type of fast slow, we're going from this more even communities through a state of being intermediate, and by past 6, past 9, we're getting into this Chrysobacterium dominated state. In past 5, we had P. mosseli, this blue guy, was introduced to these communities as an invader. So what actually changed? We can see the composition changed and the bacteria actually changed. Well, there's some things that we like to measure when we're looking at evolutionary change in bacteria. growth rates, in terms of, we can look at host association. What we found is, so host association might be relevant, so we measure that first, found that literally every possible change occurred. So M. oxidans, which was retained, so we got single species here. So O is ancestor, 1, 2, 3 are the single species passages, or they're with worms, but there's no other bacteria. And then A1 and I2 are communities. These are two community-based isolates. M. oxidans in single species became a slightly better colonizer. In communities, not so much, but communities can slow things down. So maybe that's what happened. Our pseudomonad became a worse monocolonizer over time. And our other two bacteria, so Arthropacter was interesting because it got excluded. We only have single species passages here. Didn't change. Chrysobacterium, which is most of our populations, also didn't change. Literally, better or worse, no change. Every combination, every possible direction of adaptive change was observed. And then in terms of things we'd like to measure, in vitro growth, carrying capacity, nothing. None of these are significant. Again, this is something that we see canonically. The things that we like to measure about bacteria are not the things that bacteria actually care about during adaptation. And Lance is nodding at me. But the things that do change, I think they are interested in, or how they interact with space and how they interact with each other. So these are our pseudomonad. It's obviously very motile. Both of these are more motile than their ancestor, and obviously one of these is much more motile than the other. So motility changed really dramatically in these isolates. And it turns out that there are differences in invasibility. Which isolates you had, the motility of those isolates changed how invasible you were. So in this case, there's Arthropacter, which is excluded early on. We used it as an invader to see, can we exclude it again? The ancestral community, so community-based on composed of entirely ancestor isolates, did a great job of excluding the ancestral Arthropacter. The adapted Arthropacter, not so much. The adapted communities, so the adapted community A1, Pas9, I2, Pas9, did worse at excluding the ancestral Arthropacter, but better at excluding the evolved Arthropacter. So we have changes that's happened over time that influence the ecology, influence the invasibility specifically of these communities. So when we tried doing annotation, we got what we expected in a microbial community in some ways, some interesting insights into what's actually changing over time. This fast, slow dynamic, which we expect. We have convergence in composition, where traits are a factor. Allelopathy was driving at least some of this, but it's concealing a lot of variation in the traits of the actual species, and there's ecological consequences to those traits. We had an invader here, and full disclosure, we didn't plan the invader; it just happened. So, what happens if we start over without the invader? So we're going to do this again. Same setup, same communities. This time, no invaders, no surprises. We get the same thing in terms of the broad tracks. We have this fast, slow dynamic, Pas1's up here, Pas2, Pas3, and by like Pas5, we're in this range. But noticeably, there's a range. Like sometimes some of our communities are up here, some of our communities are down here. That's true for the rest of our passes. And this is why. We have two chrysobacteria. Remember, we have CG, specifically Chrysobacterium glium, which is a very aggressive allelopath, and C. endologenes, which is less so. By even Pas5, you can see kind of a banding pattern. 4 with a lot of yellow, 4 with a lot of green, 4 with a lot of yellow, 4 with a lot of green. A lot of yellow is Chrysobacterium glium. That's our more aggressive allelopath. And that's pretty consistent. See, the less aggressive allelopath takes a smaller fraction of the community. We get more Ochrebacter, more microbacteria. If we have a more aggressive allelopath, we get a lot of allelopath. Also, we did this outside the worms. So same passage, same patching scheme, same everything, except we just didn't put any worms on the plates. And we get the same fast, slow dynamics, we get the same convergence, but the composition is totally different. So whatever's happening here, the worm is playing an important role. These are very, very different communities. And this time, we start to see variation within the species. So not only are we having variation across communities in terms of the composition to this banding pattern, we're having variation within the taxa. Specifically, what we saw is in Chrysobacterium and Microbacterium, we have morphs emerging on the place. This is visible variation. And this is what this looks like. So I'm just going to show you procedurally, that's where we're going to focus. This big guy here, this is our original morph. It's big, it's goopy, it's shiny, it's safety orange. The alternate morphs right here, a lot flatter, colonies a lot lower. We can see the rings more clearly. It's still glossy, and it's not. but it's much smaller, it's much flatter, the color is much darker, suggesting that there's some sort of differences in maybe gas production. There's something that's changing the properties of the colony. Now, C. blium is a swarm. This is another motile bacteria. And so here we were looking at swarms of these guys on swarming agar, swarming NGM agars, nematode growth medium, which we do these experiments. And we can see, so this is ancestral. We can see, it's kind of center of the swarm, and we've got these kind of lobes, like pushing out lobes as the colony starts to swarm outwards. This is a original type isolate from community F1 from the 10th pass of these communities. We see it's still very motile, but the pattern of motility has changed a little bit. We don't have this nice kind of regular big lobes. We have more regular lobing around the outside, suggesting there's been some changes in the specifics of motility. In fact, we see a lot of changes in the specifics of motility. In particular, one glaring difference that we see is the ancestral isolates are much less motile. We have much less spreading on the surfaces of plates. And this is despite growth rates being essentially the same. So once again, we're seeing differences in motility that have emerged. Now, that's about as much as I'm going to show you there, because we're still processing a lot of those data. But so we've got over what we've learned using this system. We use the worm to assemble simple gut communities. They have characteristic ecology. Structure and the function of these communities changes coherently during adaptation. There's rules that are being followed. There are patterns, and there's a lot of variation that comes with those patterns. These are not deterministic patterns. These are patterns of variation. And in terms of what we see in selection, we see most of the things that we care about bacteria, growth rates, carrying capacities in the media. These are not the things that they care about. These are not the things that are changing. What's changing is how they interact with space, how they interact with one another. All right, so I'm going to leave there to leave time for questions, or thank that group again, and I'll take your questions at this time.
So at the beginning, you mentioned the worms will eat whatever fits. So did you see like some kind of association with the size of the bacteria, the physical size, and how well they were able to survive?
So for these bacteria, they're all actually pretty similar sized. None of these are particularly large cells. We did see a difference. So the things that seem to make a difference when they're colonizing in liquid culture are if you restrict aerobe, you don't do very well. The inside of the worm is somewhat microaerobic, bacillus, the swingomonad these are aerobic bacteria. They don't in general do particularly well. On plates, the situation changed. We actually saw during one of the rounds of evolution of the single species plates, bacillus is it's not a very good colonizer of the worm in general, but it is a very good biofilm form. And so they developed thicker biofilms, heavier biofilms on the plates, we think to avoid predation. So there are, not in this set, I'm sure if we had like very different size cells, very different shaped cells, perhaps we would see more distinct effects of that. Because the shape of the pharynx turns out is important for ingestion and efficiency. In this set, that doesn't seem to be a major factor, but in terms of the structures they're able to form, now is when we start to get effect, particularly in physically structured environments. So we do think that's part of what's going on in these evolution experiments is because we're feeding them on plates, and so when we're seeing adaptation. Again, particularly for chrysobacterium, the chrysobacterium is not an awesome colonizer of the worms. And we go back a little bit, find that. It's not, again, so this is C. blium. It's not an awesome colonizer on its own, just like the per worm load is low. And it is a frank pathogen of the worms. And you see that on the plates, the worm communities on these plates. Are smaller, they're struggling, we have very few full-sized adults, there's substantial mortality. Now, if they killed all the worms, the plate would have stopped. You have to have a worm community at the end of the passage to go into the next passage. They didn't kill all the worms, but we do see a lot of pathogenicity. And so pathogenicity also didn't change But it's entirely plausible that that at least part of what's changing is the interaction on the plate, that they're reacting as a predator, right? As prey or predator rather than as a microbe, as a passenger microbe primarily. That's some of the experiments we're doing now.
Do you have any plans to do any like genomics on these bacteria?
Yes. So I didn't show the genomics from the first round. We have some genomics there. And so we did some sequencing of these guys, like the pseudomonas that came in. A lot of it was unannotated genes, like you do, but there was a flagella gene that got changed. And in the arthobacters, one of these arthobacters is much better invader. We gave us an isolate that was much more invasive, much better as an invader than the ancestor. That one we did genomics on as well, and I don't know if I actually saw the slide. We saw a lot of stress response genes. So suggesting that again, maybe we're dealing with allelopathy, maybe we're dealing with it doesn't colonize any better, so it doesn't seem like it's better able to withstand the stress of the host specifically. Again, the obvious theory is that it's interactions. It's changing into how it responds to interactions with other members. And for the second round, we've got these changes in motility. We've sequenced some of these guys, and it literally got the Dropbox like this morning. So I'll let you know.
Did you sequence like the entire population? You were able to get overall?
No, we did not sequence the entire population. That would be interesting. If I can convince NSF to let me do it, then I will. I think it'd be really interesting to see what the total diversity is. But this has been isolates. So we've been doing isolate picks, and we do see diversity among isolates, even within morphotypes. In terms of, again, like we can see some of that here. In terms of there are, even within types, so even within alternate morphs, there's a lot of variation in motility among original morphs. Some of them are much more motile than ancestor, some become less motile. We're getting more data on this right now again, but it's half of this is in my master student's notebook at the moment, which is why you're not seeing it. But you know, we do see variation across isolates, even within the same kind of morphs. Just that morph was the obvious like visual indicator that there's diversity. There's obviously a great deal of diversity in here that we are not seeing, and I'm sure there's diversity in here in the other community members that did not show up as morphotypes, so we haven't described. I think it'd be really interesting.
Oh, I'm just curious, like, what is the carrying capacity like that genomics that's used?
So you can do this kind of back of the envelope. Assume the worm is a cylinder. And you end up with a carrying capacity for E. coli of about 10 to the 5 CFU per worm, and that is pretty close to what we see for a good colonizer. Maximum capacity close to 10 to the 5. A pathogen that's growing out of control can go up to 10 to the 6, but the worm is like clearly distended at that point, and they're just very unhappy with life. So about 10 to the 5 seems to be like maximum comfortable limit for a worm-sized object with roughly E. coli-sized bacteria.
Does that impact like their ability to like eat and stuff, like absorb nutrients?
So it doesn't seem to, interestingly, and again, part of it is just the nature of the communities where worm feeding is weird. It's kind of first answer there. So worms are really, really processive feeders. We measured this at some point. I read this and didn't believe it. I measured it an adult N2 C. elegans, young day 2 adult feeding ad libitum in liquid culture can get through like 10 to the 6 bacteria per hour. They can plow through bacteria. And the defecation cycle is also really stereotyped. Most things in C. elegans are stereotyped, right? They're running on automatic in a lot of their physiology, which is again very convenient. So the stereotype defecation cycle is for wild type worm, that they'll kind of pulse forward, pulse back, and expel like roughly a third of the gut contents about once every 42 plus or minus 5 seconds. Very stereotype motor program, but that does slow down. slows down with age. It does seem to slow down with bacterial burden. So plausibly, the bacteria in the gut are slow, either mechanically slowing the peristalsis, or there's some signaling that's happening that's slowing the peristalsis. And what calling peristalsis, they're a millimeter long. How much is that peristalsis? I don't know. But it does seem to change the motor function, it does seem to change the pattern of motility. They seem to be doing fine in terms of getting nutrients. So it's not obvious that that interferes with nutrient production or nutrient absorption, and it does seem to be the case that some of these bacteria have at least a lot of capacity to produce things that the worm benefits from, specifically vitamins, cofactors. But we have one paper from my postdoc where we colonized worms with P. cellulosa, cellulitic bacteria, and gave them the ability to get carbon from cellulose. So we can have breakdown of complex compounds by the microbiome and then absorption by the host. So there might be a trade-off there as well in terms of like how much direct absorption are you losing versus how much community processed absorption are you gaining from the microbial load.
All right, I have more questions about future research. Do you have any plans to like do experiments where you're like modifying the environment of the host?
Yes.
And like, is this a feasible system to do that with?
Yes, so worms are great actually for modifying the environment. They're very tolerant to a range of environments. There's some things they don't do well with, like high temperature. There is kind of a temperature lid for worms. They get temperature stressed past like 25 degrees Celsius, and below 12, they slow down a lot. But there's a lot of things they really don't care. So as long as they're in like osmolarity is something that they don't care very much about. They have a lot of mechanisms for tolerating osmotic shock. So we have them in a specific like salt environment, but you can give them less salt or more salt pretty easily. They don't desiccate extremely well, but you can have them in a more desiccated environment for a period of time as long as you return them. They're fantastic for oxygen stress. You can put them in the anaerobic chamber for a day, but fine, which is kind of wild. So the conditions that we're thinking of looking at are mostly nutritional conditions, where we've done antibiotics, and that's really easy to do. We're looking right now at nutritional environments, periodically changing or providing bursts of nutrition for the host or for its microbes, and seeing what effects that has on the community, both as like kind of two sets of projects in terms of what are we feeding the host? Can we provide them with a nutrient like a prebiotic to change the structure or function of the community? And we started using them as a transmission model. So if we have, in this case, we have worms that are eating bacteria. Those bacteria can then get passage into the environment. We expel live bacteria, which can then grow in the environment. And it turns out, like, went through a lot the way people usually estimate excretion rate does not work because they farm. So what happens is they'll poop out live bacteria, and then they'll be eating in the same area, and they'll just drag the colony around. So I don't know how many excretion events that is. It's at least one. It also might be 20. I don't know. So you can use this. You can do like some like binomial statistics on this with the number of empty plates to get to an estimate of excretion rate, but you can't actually get a point estimate of excretion rate doing this. That's worth much of anything. But they do excrete bacteria into the environment. If you do a better experiment than this, you have better experiment than this, you can measure the rate of excretion. And you can get the rate, you can get the variance. It turns out it's very describable. It's no distributions. So we can use this then as a transmission model, because if we're defecating into an environment and then proliferating that environment, we can change the properties of the external pool, put new susceptible worms into it, and see what transmission looks like. And so we have a number of experiments like that I can talk about today. That's ongoing. Thanks.
So for the data you show of the changes over time, so that really looks at the dynamic aspect of the microbiome. I was just wondering, were those only exposed once to those specific compositions, or was it continuous?
So these are continuous, and the reason for that is that we're trying to use a simple model where migration is a constant, and so for that we force migration to be a constant. We have done some experiments with removal of migration and with tuning migration rates, so changing migration rates from like phase 1 to phase 2, and looked at the effects on the community. I don't know if I have those data, but so essentially, certainly for the bacteria that grow like lockable terra, where we have the colonization phase, the exponential phase, and saturation phase. Change in the migration rate does basically nothing after initial colonization. Once they're colonized, they just grow. And that's what should happen, right? Exponential growth should take over, and it does. It's not homogeneous though. There are some worms, there are some worms that are actually on lockable terra, and there are some worms that are not, and they'll go down. We couldn't see that. We can't see that when you keep migration rate on, but we can compare plus and minus migration and get from that. We're starting to get us what's the distribution of rates across the population. We can colonize them and then withdraw colonies and then withdraw migrants. And so that's another perturbation that we've been using. And again, we see a lot of heterogeneity in terms of the response. Some things doesn't matter. They're following lockable terra, and once you're on the curve, you're on the curve, and you're past small number effects. The stochastic effects basically just go away. You're large numbers. For some things you don't, and that's different individuals within the same population. It seems like the fraction is really stable. So we can describe this as a distribution of rates across the population. Yeah, for these experiments, we wanted a simple model with migration as constant, so of course migration be constant.